Thank you so much for the invitation to speak. Very excited to be here and tell the story of Optigo Biotherapeutics. This is actually perfect timing in terms of the order of our presentation, following so many other companies that have addressed the very same issue of the injection frequency of anti-VEGF drugs and other intravitreal biologic treatments. We're taking a similar but very different approach. And as you'll see, you'll understand why we believe our approach is quite elegant, straightforward, and lacks a lot of the complications that some of the other therapeutics that you've heard about today may face. And so our goal at Optigo is to develop safer, more convenient treatments for neovascular and degenerative retinal diseases that doesn't compromise on efficacy or safety. I don't need to educate you again about the market size and the unmet need. One thing I do want to point out is that while we are initially focusing on retinal neovascular diseases with our anti-VEGF based on aflibercept, we do have a platform that we've created that is compatible with anti-complement as well as many other classes of intravitreal biologics that we'd like to pursue in the future. Right now we're raising a Series A $30 million financing that will get us through IND as well as our first-in-human phase 2 trial. We have broad global IP on our platform as well as our lead compound going through 2044 with great data exclusivity and a very, very experienced team that's done this for a long time. What is our goal? Is to take existing best-in-class anti-VEGF today, which is aflibercept, and take it from once every 1 to 3 months at best and extend it to every 6 to 9 or even 12 months. And we'll talk about how. The reason why we believe reducing injection frequency is important is that it's not just a matter of convenience. That injection frequency leads to high treatment burden. That high treatment burden leads to poor adherence. And that poor adherence, as all of the studies have shown, lead to poor outcomes. And so in the real world, you do not see the same efficacy that you see in the phase 3 trials because patients in the real world are never able to get the access to drug and or retina specialists that you do in a very tightly controlled phase 3 trial. So our aim is to keep the safety and efficacy of existing best-in-class anti-VEGF drugs while extending the duration to, again, to that sweet spot of 6 to 9 months. So how are we achieving that? As you've heard other companies talk about, they're injecting polymers into the eye, right? So you're hearing about hyaluronic acid, other hydrogels, et cetera. What are we doing that's different? We're actually taking advantage of a hydrogel polymer that Mother Nature already put in our eyes, namely the vitreous gel, and namely 3 different components of the vitreous gel, type 2 collagen, albumin, and hyaluronic acid, and leveraging those as our targets, as our anchors, in order to anchor our aflibercept-based molecule 2, with the idea that if we can anchor to one of those 3 components of the vitreous with tight enough efficiency so that it binds, but not too tight that it's forming a depot and doesn't actually go to the retina and bind VEGF, we can actually extend the half-life dramatically. And so what we've done over the last 5 years, and this has been a gargantuan effort over a long time with many people involved, is to take an existing therapeutic, as you can see in gray, add a proprietary anchoring ligand, which we have against each of those 3 different molecule targets in the vitreous. And then here we have aflibercept as our first enablement of this. We can use, again, almost any biologic drug and test at each of these. And what we've shown is that we can extend the half-life of intravitreal drugs. We get new composition of IP, a composition of matter IP with each of our constructs. And our lead asset shows that we can get at least 3 times increase in the half-life versus Eylea. And let's talk through some data. We've done rabbit efficacy prolongation and PK prolongation studies where the drug is injected through a regular needle into the eye, no different than aflibercept or Eylea or any of the other anti-VEGFs are. We have no fancy injection mechanisms, no polymers, no high viscosity, large pellets, anything. This is a standard injection like the retina doctors are used to. And in both the efficacy and PK platforms, and I'm deliberately not showing the actual molecules and targets involved just because we have that for our poster on Tuesday. So I encourage you to come on Tuesday, or we'll show you the poster separately today, but it's embargoed until our official presentation. But suffice it to say that we have our lead and our second backup molecule can extend the half-life 3 to 4 times, both in efficacy and in the pharmacokinetic study in rabbit, with allometric scaling to humans showing that the 2 milligram equivalent, which for us is 2.5 milligrams of aflibercept-based drug, can prolong injection frequency to at least 6 months. We think we can go dramatically higher. No inflammation seen in histopathology examinations, which is very important. And again, we have composition of matter filed with exclusivity till at least 2044. This is what some of our data looks like, right? Well, we went is head-to-head against aflibercept, and as you can see, that by day 12, you get a lot of leakage with a score of 3 out of 4 with aflibercept. It jumps to 4 by day 28. With our molecule, it stays at 0 score of leakage on day 12, and it's not until day 28 that you start seeing a little bit of leakage, and it gets a little bit worse by day 42. And this is in a rabbit, so we have clear differentiation here compared to the standard of care. What's our roadmap? So far, since 2020, so in the last 5.5, almost 6 years, we've done a lot of drug design in CMC, a lot of non-clinical studies in rat and rabbit. Right now, we're doing GMP-like manufacture, clean product to go into a pivotal NHP study. We're planning to have that wrapped up very shortly, and then we will initiate, in a 17-month process, our path to IND, and from 18 months after that, we hope to have our phase 2 data well controlled against the aflibercept so that we really can prove our prolongation of duration in the real world rather than just hypothetically. We were going to require about $30 million to get to that point, and after that we can of course go into studies in multiple indications for approval. Very experienced team. Our CEO, Ardekani, is here in the audience, so you're welcome to come talk to any of us. We have Udo Vetter, who does the manufacturing for many of the intravitreal and systemic and sterile injectable biologics involved with us as an investor and board advisor, and a very accomplished board. We have great advisors and KOLs advising us, including Gary Novak for regulatory, David Boyer for clinical, and so many others who are ex-FDA and have been involved in the approval of biologics. So with that, I'll thank you very much.
Thank you. Just have time for maybe one question. I have a question. It's been a while since I've done VEGF challenge, but I thought aflibercept typically lasted longer than 12 days. I remember ranibizumab lasting about that long, but Eylea seemed to be longer in general. Is it a lower dose of aflibercept that you're using in the standard trials?
Yeah, that's a very good question. What we wanted to do is actually tweak this model in order to be able to get data pretty quickly. So what we did is we titrated our VEGF dose, and we did a lot of different studies to figure out what is the lower limit of VEGF that you need to put in to get leakage reliably across all the animals. That way, we can put in the minimum amount of anti-VEGF, and this way you get much shorter studies. Otherwise, we've been waiting forever for data.
Got it.
Good question.
Thanks very much.
Great. Thank you.
Thank you.